About the mechanisms of cell injury and the types of cell injury. We discussed about the reversible and irreversible injury. So when there is irreversible injury, ultimately it will lead to cell death. So when we discuss the modes of cell death, it can be either non-programmed cell death or programmed cell death. And in the programmed cell death also we have the apoptotic cell death or non-apoptotic cell death. So in the non-programmed cell death, we have only one entity, that is necrosis. In the programmed apoptotic cell death, we have two mechanisms, in which the major one is the apoptosis and the other one is NETosis. And in the programmed non-apoptotic cell death, there are various modalities. One of them is the vacuole forming mode of cell death, of which the autophagy is the most important one, the others being NETosis, MITOSIS and Pyroptosis. The mitochondrial-dependent cell death, that is Mitochondrial-dependent and Pathology. The iron-dependent cell death, that is Phyroptosis. The immune-mediated mode of cell death, that includes Pyroptosis and NETosis, and the others include Necroptosis. So for coming first to the non-programmed cell death, that is necrosis. So what is necrosis? Necrosis is defined as a localized area of death of tissue in which there is contiguous death of several cells, and that is characterized by what? There is denaturation of the cellular proteins, means the mechanisms which led to irreversible cell injury that are functional in necrosis also. There is denaturation of cellular proteins, then leakage of the cellular contents through the damaged membranes. Now, because there is leakage of the cellular contents, there will be inflammation, and lastly, there will be enzymatic digestion of the lethally injured cell. The enzymatic digestion is because the body needs to clear this lethally injured cells to curb more damage. So ultimately, there is enzymatic digestion of the lethally injured cells. So why there is inflammation in case of necrosis? The severe damage to the membranes, this leads to leakage of the cellular contents. These are called as the damage associated molecular patterns, like the ATP, which is released from the mitochondria, uric acid, which is released from the nuclear breakdown. Now, when these are released into the extracellular space, these are recognized by the phagocytes or the macrophages as foreign body. Now, macrophages are the cells which eat up anything and just clear the debris. So they recognize these substances as extraneous material, and then there is phagocytosis of this debris, and this leads to clearance of the necrotic cell. Now these molecules, these macrophages, this also leads to the production of various cytokines that induces inflammation, and these cytokines also release some proteolytic enzymes to ultimately clear the necrotic cells. So that's why necrosis is characterized by inflammation. Now, what are the patterns of tissue necrosis? There are 5 basic patterns of tissue necrosis. First and foremost, coagulative necrosis. So what is coagulative necrosis? The most important cause is basically ischemia due to any obstruction in the vessel, and a localized area of coagulative necrosis that is called as an infarct. In this, the architecture of the dead tissue is usually preserved, and there is a firm texture of the tissue. Now, how the architecture of the dead tissue is preserved? Now, because of injury, there is denaturation of not only the structural proteins, which leads to cell injury, but also there are enzymes which blocks the proteolysis of the dead cells. The enzymes which cause proteolysis, that are also denatured in this process. So the cell morphology is preserved for some days. And what we see in morphology, if we compare the right with the left aspect, the left aspect is showing the normal tubular basement membrane. We can see the nucleus is well preserved. While the right aspect, we can see there is increased eosinophilia of the cytoplasm and the nucleus is absent. There is complete karyolysis. However, a ghost outline of the tubules is seen. This is called as tombstone appearance. And also there is surrounding inflammation. The surrounding inflammation will clear up the necrotic cell. This gross photomicrograph is showing a wedge-shaped infarct in case of kidney. So next, coming to the next modality, that is liquefactive necrosis. The term liquefactive necrosis is usually applied in case of central nervous system. This is also a sort of coagulative necrosis, but in central nervous system. In this, there is transformation of the tissue into a liquid viscous mass. The brain is already very soft in appearance. So what happens is there, because of bacterial or fungal infection or any microbial insult, there's accumulation of these leukocytes and liberation of enzymes from this cell. So ultimately there is accumulation of inflammatory cells like neutrophils, and this liquid viscous mass is called as pus. And in photomicrograph, we can see that this is the normal appearance of the brain. This is the center of liquefactive necrosis, where we can say a cystic cavity with this debris-like material. Next, gangrenous necrosis. This is not a specific pattern of cell death. It is a subtype of coagulative necrosis, usually applied to a limb like the lower leg, when it has lost its blood supply and undergone necrosis. Now, what happens when there is superadded bacterial infection? This causes liquefactive necrosis because of the action of degradative enzymes in the bacteria and the attracted leukocytes. This gives rise to what is called as the wet gangrene. Now, the next important modality is the caseous necrosis. The caseous necrosis term is used almost synonymously with tuberculosis. When there is tubercular infection, what we see in morphology is a granuloma formation. So in granuloma, the granuloma is characterized by a central area of caseous necrosis, which appears powdery white in gross. And this central area of caseous necrosis that is surrounded by epithelioid macrophages, Langhans type of giant cells, and lymphocytes. Next, fibrinoid necrosis. Now, fibrinoid necrosis is usually because of immune reaction, that is antigen-antibody reaction. This is the prototype is vasculitis, where we can see the antigen antibody deposition in the vessel wall causes damage of the vessel wall along with the exuded fibrin material. So that is called as the fibrinoid necrosis. Next, fat necrosis. The fat necrosis, this term is used when mainly in fatty tissues or in pancreas or breast. What happens is when there is acute pancreatitis, this causes a release of activated pancreatic lipases into the omental fat or in the pancreas itself. Now this the fatty acids generated because of these lipases, this combines with calcium to form this grossly visible chalky white deposits, which is called as the fat saponification. And what we see in histology, these are the normal adipocytes, while the remaining adipocytes have been digested. There are some ghost outlines. Along with this, we can see lot of macrophages, which will try to clear off the debris and inflammatory cells. And when this is long standing, then there will be dystrophic calcification. What we see in necrosis, so after reversible injury, either there will be recovery, or if there is progressive injury, there will be irreversible injury, and finally necrosis. The morphological features are like that of irreversible injury, that is swelling of the endoplasmic reticulum or mitochondria, the membrane blebs, then nuclear breakdown like karyorrhexis, pycnosis, karyolysis, amorphous densities in mitochondria. So this occurs less frequently and involves many cells. This is an abnormal and uncontrolled cell death, and mostly it is associated with pathological condition. Whenever there is necrosis, we must suspect that there is underlying pathological condition only. There is no physiological cause of necrosis, and be caused by any injurious stimuli. This is caspase independent. Now this caspase we will learn in apoptosis. The pathway of apoptosis depends on caspase. However, necrosis is a caspase independent pathway. Inflammation is always present, and in the morphology we will see that there's cell swelling and the cell will burst. There will be swelling of the mitochondria and endoplasmic reticulum. There is leakage and enzymatic digestion of the neighboring cellular contents. And then there will be disruption of the membranes, the plasma membranes, and there will be increased eosinophilia and nuclear changes. Now coming to the next important modality, that is the programmed apoptotic cell death, the major modality being apoptosis. So what is apoptosis? The apoptosis is a type of cell death that is induced by tightly regulated suicide program. in which the cells which are destined to die, this activate the intrinsic enzymes that degrade the cell's genomic DNA and nuclear and cytoplasmic proteins. Now, what happens in human body? There is continuous turnover of cells. It is said that there is a turnover of about million cells in a second. So all the cells won't be useful in our body. So these cells need to be removed, because if they are not removed, then this will lead to unnecessary growth, which is called as neoplasia. And also the cells can accumulate mutant protein. So this excess cells, this needs to be removed, and the process of removal of these cells is by a process which is called as apoptosis. In this is characterized by formation of apoptotic bodies. So what happens is this: from intact cell, there is formation of these apoptotic bodies. The membranes remain intact, and this apoptotic bodies comprises of fragments of cytoplasm, some fragments of nucleus, and cellular organelles. Now, as this as the cytoplasmic membranes and other membranes remain intact in this process, the contents are not released into the circulation, so inflammatory reaction is not present in case of apoptosis, unlike necrosis, where there is lysis of the cell, and this provokes the release of cytokines and inflammatory response. So this is one point of difference between apoptosis and necrosis. While apoptosis is not associated with inflammation, necrosis is associated with inflammation. Now, what are the causes of apoptosis? In case of necrosis, we have seen that this is associated almost always with pathological conditions, whereas in apoptosis, it can be present in physiological situations as well as in pathological conditions. Now, what are the physiological situations which can be associated with apoptosis? The normal embryogenesis. In this case, the primordial structures, this needs to be removed. So this removal is helped by the process of apoptosis. Involution of hormone-dependent tissues upon hormone withdrawal. In case of lactating breast, the atrophy of the lactating breast, that is brought about by the process of apoptosis after weaning. Then cell loss in proliferating cell populations. The proliferating cell populations in our body include the epithelial cells in the GIT, and also some immature cells like in the thymus and bone marrow. So these needs to be removed. So this is removed by a process called as apoptosis. Elimination of potentially harmful self-reactive lymphocytes. Now this harmful self-reactive lymphocytes also need to be removed so that they do not cause autoimmune reactions in our body. That is also done by apoptosis. And death of the host cells that have served their useful purpose, like macrophages and neutrophils. Their purpose is to clear out the microbial insult. So when they have served their useful purpose, they need to be removed so that they do not release the harmful cytokines which are harmful for the body. So this is also brought about by apoptosis. Now these are the physiological conditions. Now what are the pathological conditions? Suppose there is DNA damage because of radiation, anti-cancer drugs, hypoxia, or whatever. Now this damaged cells also needs to be removed, because if these mutant cells are present in our body, they can give rise to neoplasia. So this removal is done by apoptosis. Accumulation of misfolded proteins. Accumulation of misfolded proteins causes various diseases like the prion diseases or the cystic fibrosis, etc. The last class I discussed about the endoplasmic reticulum stress, which is due to misfolded proteins. So this removal of misfolded proteins, that is also done by the process of apoptosis. Certain infections, especially virally infected cells, like in hepatitis or HIV, the virally infected cells are removed by the process of apoptosis. And pathological atrophy in parenchymal organs after duct obstruction, like in salivary gland duct obstruction or pancreatic duct obstruction, the pathological atrophy, that is also brought about by the process of apoptosis. Now, what are the morphological and biochemical changes in apoptosis? While necrosis was characterized by cell swelling, this apoptosis is characterized by cell shrinkage. There is chromatin condensation and there is no nuclear fragmentation. The nucleus remains intact, though it can be converted, whatever is engulfed, that is intact. It breaks into some fragments and that is engulfed into apoptotic body, but there is no caryolysis in this process of apoptosis. And ultimately, there's phagocytosis of the apoptotic cells by macrophages. So this is a photomicrograph which is showing this is squamous epithelium, where we can see this is a apoptotic body. This is a cell which is showing increased eosinophilia, and there is chromatin condensation as compared to this nucleus. And in electron micrograph also we can see this condensed chromatin under the nuclear membrane. Now, what are the biochemical features? The process of apoptosis is characterized by activation of caspases. So this is also called caspase mediated programmed cell death. So from where this term caspase, what is derived? The word C, that refers to a cysteine protease. That is an enzyme with cysteine in its active site. And aspase, this refers to unique ability of these enzymes to cleave after aspartic acid residues. So, so the caspase term is derived from this. Now, there are two types of caspases: initiator caspase and executioner caspase. The initiator caspases are caspase 8 and caspase 9, while the executioner caspases are caspase 3 and caspase 6. Another thing which we see in apoptosis is DNA and protein breakdown that can be found out by the process of electrophoresis. So what we find in electrophoresis is a method of separating out protein fragments based on their charge and also based on their molecular weight. This is an intact cell where the DNA is intact. Now, in the process of apoptosis, by the activation of endonuclease, there is breakdown of DNA into 50 to 300 kilobase pieces, which are further broken down into 180 to 2000 base pairs. So this can be seen in electrophoresis as separated out DNA fragments. This is called as the ladder pattern. And in case of necrosis, we will see complete smearing of this DNA fragments. Now, what are the membrane alterations seen? Now, what happens in the process of apoptosis? After the process of apoptosis is completed, these apoptotic cells are cleared by macrophages. So, to make the cell palatable to the macrophages, there is some alteration in the phospholipid membrane. Now, phosphatidylserine, this is usually located in the inner leaflet of the plasma membrane. In case of apoptosis, the apoptotic body, there is translocation of this phosphatidylserine from the inner aspect to the outer aspect, so that the macrophage can identify the apoptotic cell and can clear out the cell. So these lipids are detectable by a protein which is called as annexin 5, and this annexin 5 staining is used to identify the apoptotic cells. Now, coming to the mechanism of apoptosis. The process of apoptosis is divided into two phases, and there are two pathways. The phases are an initiation phase where there is activation of the caspases, and the execution phase where the terminal caspases this trigger cellular fragmentation. And also there are two pathways: the intrinsic pathway or the mitochondrial pathway, which is the major pathway, and the extrinsic or the death receptor initiated pathway. So coming to the intrinsic or the mitochondrial pathway, this is the major mechanism of apoptosis. This results from increased mitochondrial permeability and release of pro-apoptotic molecules into the cytoplasm. Now, this mitochondria contains substances like cytochrome C. Now, this cytochrome C molecule is like a double-edged sword. Now, on one hand, it is required for the generation of ATP, and ATP is required for various synthetic functions. On the other hand, this cytochrome C, when it is released from the mitochondrial membrane into the cytoplasm, this initiates the suicidal program of apoptosis. Now, the release of these mitochondrial proteins is also controlled by a finely orchestrated balance between the pro-apoptotic molecules and the anti-apoptotic members, and this belongs to the family BCL2 family of proteins. Now, coming to the BCL2 family of proteins, these are divided based on their pro-apoptotic, that is, the molecules which will enhance apoptosis, or anti-apoptotic functions, and this is based on the BCL2 homology domains they possess. Now, the BCL2 the BCL2 gene, this derived from the name B cell lymphoma. The B cell gene is altered in various subtypes of B cell lymphomas. So the name is derived from there. So what are the anti-apoptotic proteins? Mean the proteins which inhibits apoptosis. BCL2, BCLX and MCL1. You should remember the example of this. These are sometimes asked in your exams also. Now, what is the function of this anti-apoptotic gene? They keep the mitochondrial outer membrane impermeable. So thereby, they are preventing the leakage of the cytochrome C and other death-inducing proteins into the cytosol. What are the pro-apoptotic genes? BAX and BAC. Now, this has 1 to 3 domains of this BH. So this is what is the function of this pro-apoptotic genes? The pro-apoptotic genes form a channel in the outer mitochondrial membrane, and this allows the leakage of cytochrome C from the mitochondrial membrane into the cytosol, and this will initiate apoptosis. Now, there are some regulators also. The regulated apoptosis initiators. These are also in a way pro-apoptotic genes, and these are BIM, WID, BAD, PUMA and NOXA. These are also called BH3 only proteins because this contains only the third domain of the BH3 gene. And when these are upregulated or activated by stress, they can also initiate apoptosis. Now coming to the mechanism of apoptosis. When there is lack of survival signal or DNA damage, there will be decrease in the anti-apoptotic genes and there will be activation of the pro-apoptotic genes. Now what happens in a viable cell? When the viable cell receives adequate survival signal like the growth factor, there will be production of the anti-apoptotic proteins like BCL2. So this will prevent this will keep the mitochondrial membrane intact and there will be no leakage of the cytochrome C into the cytoplasm. So the cell will be intact. Now, when there is lack of survival signals or there is DNA damage because of irradiation or any toxin or anything, there will be activation of the sensors, that is BH3 only proteins. So this BH3 only proteins, in one hand, this will antagonize the function of the anti-apoptotic genes like BCL2. On the other hand, it will also activate the BAX-BAC channel, which are pro-apoptotic genes. So this will ultimately cause the leakage of cytochrome C and other proteins. So when there is leakage of cytochrome C, this cytochrome C binds to a protein which is also called as the apoptosis activating factor 1, and this is called as apoptosome. Now this structure, this will bind to pro-caspase 9, that is the inactivated stage of the caspase 9, and will convert it into an active caspase 9. These are the initiator caspases of the mitochondrial pathways. Now this initiator caspase will activate the execution caspase, that is it will convert the pro-caspase 3 into caspase 3, and this will bring about apoptosis. Now there are some inhibitors of apoptosis also which tries to inhibit the apoptosis. So this has to be inhibited by certain substances. So the mitochondria also releases some substances which are called as the SMAC or diblo. The function of this is to inhibit the inhibitor of apoptosis, so this apoptosis can progress. So the signal 1 is the generation of the cytochrome C or release of cytochrome C, and this signal 2 is in the form of release of SMAC and diblo. This can inhibit the inhibitor of apoptosis so that there can be apoptosis. So this is about the intrinsic or mitochondrial path. The other pathway is the extrinsic or the death receptor initiated pathway of apoptosis. This is initiated by engagement of plasma membrane death receptors. These death receptors belong to the TNF receptor family and they contain a cytoplasmic domain which is involved in protein interactions, and this is called as the death domain. Now what happens when this FAS interacts with the FAS ligand? There is aggregation of about 3 molecules of this FAS molecule, and this FAS molecule will in turn bind to the FAD molecule which is called as the FAS associated death domain. When this is interaction of this FAS and FAD, This will lead to activation of the procaspase 8. The procaspase 8 is the initiator caspases of the extrinsic pathway. And this will lead to activation of this procaspase 8 into active caspase 8, which will further lead to activation of the executional caspases, that is caspase 3 and 6, and this will ultimately lead to apoptosis. Now in the extrinsic pathway also there is an inhibitor which is called as the flip molecule. Now this flip molecule is also released by certain some viruses so that they can escape the apoptotic cell death. The flip molecule inhibits the procaspase 8 so that there is no apoptosis. So this flip molecule is also a target of research in case of antiviral drugs. Now coming to the phase of execution. Now after the initiation by the intrinsic and extrinsic pathway and after the activation of the initiator caspases, there is ultimately activation of the executioner caspase, that is caspase 3 and caspase 6. Now this causes this caspase 3, this cleaves the inhibitor of DNase. So when the inhibitor is cleaved, this will enzymatically active the DNase and it will cause DNA breakdown or DNA degradation. Now the other thing it does is proteolyze the structural components of the nuclear matrix, and that will cause fragmentation of the nuclei. Now when there's formation of the apoptotic body, now this apoptotic body cannot be present indefinitely in the body, because if it is present in the body, it will ultimately lyse and cause inflammatory reaction. So this apoptotic body needs to be cleared by phagocytic cells like macrophages. So this apoptotic cells releases certain signals that is called as the find me signals or eat me signals, so that the macrophages are attracted to this apoptotic body, and this apoptotic body is engulfed by the macrophages and cleared out. This process is very fast in our body, so we hardly get to see any apoptotic cells. The other is the eat me signal, in which there's some alteration in the phospholipid membrane, which I have already discussed. There's translocation of phosphatidylserine from the inner leaflet to the outer leaflet, which makes it more palatable to the macrophages, and then the macrophages eat up this apoptotic cell. So in summary, what we see in apoptosis, we have two pathways, the mitochondrial or the intrinsic pathway and the death receptor or the extrinsic pathway. It depends on the activation of caspases. We have the initiator caspases and the executioner caspase, which ultimately leads to endonuclease activation, and there's formation of this apoptotic body, which is ultimately cleared by the phagocytes. Now, what are the differences between apoptosis and necrosis? This is important for your exam point of view also. While apoptosis is a programmed and coordinated cell death, while in case of necrosis, cell death is because of degradation of tissue by hydrolytic enzymes. The causative agents, apoptosis, can be both physiological or pathological, while necrosis is almost always pathological because of hypoxia or toxins. What we see in morphology, there is no necrosis in case of apoptosis because these are cleared almost immediately by the macrophages. Inflammatory reaction is always present in case of necrosis. While in apoptosis, we get death of single cells, while in necrosis, there is death of many adjacent cells. Now, apoptosis is characterized by cell shrinkage, necrosis by cell swelling. There's formation of cytoplasmic blebs on the membrane, and necrosis is characterized by membrane disruption. There's formation of apoptotic bodies. In case of necrosis, the organelles are completely damaged. There's chromatin condensation in case of apoptosis, while in case of necrosis, there's nuclear disruption. And there's phagocytosis of apoptosis bodies by macrophages. And in this, the cell debris is cleared by macrophages. What are the molecular changes? In this, the lysosomes and organelles are intact in case of apoptosis, while in necrosis, there is breakdown. The pathways are different. In case of apoptosis, it is by the intrinsic pathway or the extrinsic pathway, while in case of necrosis, it is due to some injurious stimuli, ATP depletion, free radical injury, or membrane damage. Now, Enoikis, this is also a type of programmed cell death. This is also a subtype of apoptosis. What happens is this: in normal human body, there's cell-to-cell interaction, which is by E-cadherin, and also the cell binds to the underlying matrix by the integrin molecule. Now, suppose if there is dislodgement of the cell or detachment of the cell from the underlying matrix, in normal epithelium, the cell dies, and this process is called as enoikis. Now, in cancer, what happens if this process is impaired, this enoikis is impaired, these cells will survive even after detachment from the underlying matrix, and this will lead to cell metastasis as well as cell survival. So, enoikis is a protective mechanism which can protect us from metastasis and cell survival in case of malignancy. And this is brought about by JNK signaling pathway. Now, coming to the other mode of cell death, that is necroptosis. Now, as the name suggests, this is a hybrid of necrosis. The term necro from necrosis and optosis is from apoptosis. So why it is a hybrid? Now, what are the features which are common to both necrosis and necroptosis? There's loss of ATP in both. There's swelling of cell and organelles in both. There's generation of reactive oxygen species. There is release of lysosomal enzymes. There is rupture of plasma membrane. Now, because of all these features, necroptosis will be associated with inflammation. How it is different from necrosis? Necrosis is driven passively by toxic or ischemic injury, while this is an active pathway where it depends on cell signaling. Now, what are the features which are common to apoptosis and necroptosis? These are also triggered by signal transduction pathways, and this will ultimately lead to cell death. How is it different? Unlike apoptosis, it does not require caspase for its functioning. So it is also called as caspase independent program cell death. So what happens in case of necroptosis? This is also brought about, this has similarity with the extrinsic pathway of apoptosis. An extrinsic pathway of apoptosis, I told you, that is because of the TNF receptor. So when the TNF-R1 receptor binds with TNF, this initiates the formation of a complex, that is a complex of 4 proteins, which is called as the RIPK1 complex. RIPK1 stands for receptor initiator protein kinase 1. 2 molecules are important for this pathway, that is RIPK1 and RIPK3. Now, what happens when this RIPK1 and RIPK3 binds to the another molecule, MLKL? This causes phosphorylation of this MLKL protein. When this MLKL protein is phosphorylated, there's oligomerization of this MLKL protein, and this is translocated to the plasma membrane, and this ultimately leads to disruption of the plasma membrane, which causes tissue damage, inflammation, and ultimately cell death. Now, even if this binds to the fat protein, the caspase is not activated in the process of necroptosis. So, in this way, it is different from apoptosis. So, if one asks in your exam, what are the molecules in case of necroptosis? These are either the RIPK proteins or either the MLKL proteins. So, these are the markers of necroptosis. Now, where is necroptosis there? Useful. So necroptosis in health and disease, in physiologic conditions like during the formation of mammalian bone growth plate. And what are the pathological states? In various inflammatory conditions like hepatitis, pancreatitis, in neurodegenerative conditions like Parkinson's disease or the Huntington's disease. And also it serves as a backup mechanism. Now, certain viruses like cytomegalovirus, this can secrete caspase inhibitors. So this will avoid apoptosis. So in these viruses, necroptosis can protect us from the infection by cytomegalovirus. So this chart shows the difference and similarity between necroptosis, necrosis, and apoptosis. While necroptosis and apoptosis can both be physiological and pathological condition, necrosis is mostly pathological. Now, necroptosis and apoptosis are programmed cell death, while necrosis is unprogrammed. In necroptosis and necrosis, there is release of tissue fragments because of which there is inflammation, while in case of apoptosis, there is phagocytosis. Necroptosis and necrosis is characterized by cell swelling, while apoptosis will show cell shrinkage. The formation of apoptotic body is seen in apoptosis, while there is no apoptotic body formation in case of necroptosis or necrosis, and inflammatory response, as there is cell lysis in case of these two mechanisms, inflammatory response will be present in this, while apoptosis will not show any inflammatory response. Now coming to the immune mediated mode of cell death, that one of which is pyroptosis. Now as the name suggests, pyro means fever. So this is characterized by release of a cytokine which is interleukin 1. And this is also caspase mediated. In this, the caspase which comes into function is caspase 1. Now, pyroptosis is thought to be a mechanism by which the microbes cause the death of the infected cells, and at the same time, they also trigger local inflammation. Now, what happens? The pathogenic bacteria, they release certain substances called PAMs, that is pathogen-associated molecular patterns. Now, this is recognized by the node-like receptor, that is NLRP3, and this results in the formation of a structure which is called the inflammasome. So, pyroptosis is characterized by the formation of inflammasome. This contains the sensor protein, which is the node-like receptor, and adapter protein, and caspase in the inactive state. Now, this formation of inflammasome, this converts inactive caspase into caspase 1, and this converts the pro-interleukin 1 into interleukin 1, and this leads to acute inflammation and cell death. Now, seeing the pathway in more detail, there are two pathways. One is the canonical pathway or the conventional pathway, which I just discussed. The other is the non-canonical pathway. In the non-canonical pathway, the lipopolysaccharide, the intracellular lipopolysaccharide, this directly activates the caspases 4, 5, 11. Now, ultimately, this inflammasome formation by the canonical pathway or the caspases from the non-canonical pathway, this releases the gasdermin C-terminal. The N-terminal is released from the C-terminal. So, this gasdermin, this oligomerizes in the plasma membrane, and this leads to leakage of this various interleukins, and this leads to cell death and inflammation. Now this caspase in 11 also this causes splits of a molecule called the pannexin 1. This releases ATP, which activates a molecule called the P2X7. This causes release of potassium from the intracellular region to the extracellular region, and at the same time it can also activate the formation of inflammasome, and this causes further activation of the interleukin 1. So what are the similarities and differences between apoptosis and pyroptosis, as both are caspase dependent? While apoptosis is because of intrinsic and extrinsic pathway, the pyroptosis is characterized by the formation of inflammasome. The caspases present in case of apoptosis are 3, 6, 7, while in case of pyroptosis, it is caspase 1 in canonical pathway and caspase 11 in non-canonical pathway, which will cause release of these interleukins. This will cause persistent plasma, while in apoptosis, the plasma membrane integrity is maintained. In case of pyroptosis, there is ultimately rupture of the plasma membrane, because of which there will be inflammation, while in case of apoptosis, there will be no inflammation, there will be non-immunogenic clearance of the dead cell by phagocytosis. Now, other immune-mediated pathway of cell death, that is the NETosis, that is neutrophil extracellular trap associated cell death. Now, this is also because of microbial insult. Now, what happens when there is microbial or fungal infection? We know that the neutrophils come into action. So, what the neutrophil does is, there is this dissolution of this neutrophil chromatin, and ultimately there is lysis of the neutrophilic membrane, and this release of this intracellular or DNA chromatin, this the release, this also traps, this contains some antimicrobial substances like myeloperoxidase, neutrophil elastase, and cathepsin. So the function of this is to curb the microbial insult. So in this way, the neutrophil also dies. So this is called as the neutrophil extracellular traps. This traps the microbes and contains the microbial insult. Now the molecules which are important is this when there is a microbial infection, the neutrophil releases uh, free radicals. That is, one of them is superoxide, which is generated by the NADPH oxidase, the process of autophagy, and peptidyl arginine deaminase, that is PAD4 dependent. This causes this histone citronylation, which causes opening up of the chromatin, and ultimately this chromatin is released from the neutrophil. So this is a photomicrograph. This is an intact neutrophil, where we can see this is the cytoplasm and this is the nucleus. In case of this formation of these traps, this neutrophils releases its chromatin, which contains the microbicidal agents, and this fibrillar material, this traps the bacteria. This is an electron photomicrograph where we can see this fibrillar DNA material along with the antimicrobial substances. This has trapped the staphylococcal microbe. Now, the applications where is NETosis seen, apart from the killing pathogens, it can also be pathogenic in case of brain, like the Alzheimer's disease, in lung, various lung diseases like cystic fibrosis, in liver, in cardiovascular system, reproductive system, GIT, kidney, or musculoskeletal system. These are basically areas of research, and nowadays more and more research is going in case of cell death. Now coming to Phyroptosis. Now Phyroptosis, this is an iron dependent pathway of cell death, as the name suggests. This is a distinct form of cell death where it is triggered when there is increased intracellular levels of iron or reactive oxygen species. And this means the glutathione peroxidase system. The function of this acts as antioxidant. So it balances the reactive oxygen species. So when the amount of reactive oxygen species overwhelms this glutathione dependent antioxidant defense mechanism, there will be unchecked membrane lipid peroxidation. So this will disrupt many aspects of membrane function, like the membrane fluidity, lipid-protein interactions, ion and nutrient transport, and signaling pathways. So ultimately, this will lead to loss of membrane permeability and ultimately cell death, which will resemble necrosis. This process is regulated by specific signals, unlike necrosis, and obviously, as the name suggests, it can be prevented by reducing the iron levels. Ultrastructurally, what we can see is the most prominent feature as the loss of mitochondrial cristae and rupture of the outer mitochondrial membrane. So coming to the process of Phyroptosis, in this now ferritin is the storage form of iron, the intracellular storage form of iron. So when there is increased ferritin, there is ferritinophagy, means there is autophagy of the ferritin, there is accumulation of ferritin. So this will increase the labile iron pool. Now, iron functions as a reactive oxygen species. So this will cause the increase in polyunsaturated fatty acid oxidation, which will damage the lipid membrane. Now, other thing which happens is there is damage of the cysteine glutamate antiporter system. This causes decrease in cysteine and cysteine, and this will ultimately decrease the amount of reduced glutathione, which acts as the curbs the free radical injury. So this causes injury to this selenium, this functions of the selenium dependent lipid repair enzyme. So when this lipid repair enzyme is also damaged, this will further increase the fatty acid oxidation, which will cause further damage. So this GPX4, this is a marker of ferritinophagy. Again, where we can find this Phyroptosis, mainly this is found in various cancers and neurodegenerative disorders. Neurodegenerative disorders like the Parkinson's disease and the Huntington disease, and cancers like the hepatocellular cancer, gastric or colorectal cancer, lung cancer, the clear cell cancer, or the pancreatic cancer. Now coming to the process of autophagy. Now autophagy, as the name suggests, auto means the cell is eating up its own contents. Now this involves the delivery of cytoplasmic materials to the lysosome for degradation. Now this is also a survival mechanism whereby in stage of say stress or starvation, or in case of nutrient deprivation, the cell cannibalizes their own substances so that the cell can survive and recycle those products. And this autophagy is implicated in also physiological states like aging and exercise, as well as various pathologic processes. Now coming to types of autophagy. Now the autophagy is different from heterophagy. In heterophagy, there is engulfment of the extraneous material, while in case of auto, the cell digests its own products. So the types of autophagy are microautophagy, the chaperone-mediated autophagy, the macroautophagy, and it also can be non-selective or selective. Selective, like when there is only engulfment of the endoplasmic reticulum, it is called ER-phagy. Pexophagy, when there is autophagy of the peroxisomes. Mitophagy, when only mitochondria. Ribophagy, ribosomes. Lipophagy or xenophagy, when the pathogens are. So what happens in autophagy? When in case of stress or starvation, the cytoplasmic organelles, there's formation of this initiation complex or the nucleation complex, which is a complex of 4 proteins. This leads to a formation of phagophore. This membrane of phagophore, that is derived mostly from the endoplasmic reticulum. However, it can also be derived from the plasma membrane or also from the mitochondria. So this is the initiation. Then it will elongate, and ultimately there is formation or maturation of this complete autophagosome. This is bilayered, and this has ingested the cellular contents. Then there will be fusion with the lysosome. This lysosome will fuse with this autophagosome, and it will lyse the inner membrane along with the cellular contents, and ultimately there will be recycling of the metabolites. Now the molecule which is important in autophagy is the light chain 3, which is derived from a microtubular protein. Now what are the one I discussed, that is the macroautophagy. Now what are the other types of autophagy? Microautophagy. In microautophagy, there is no formation of any phagosome or phagophore. There is direct sequestration of the cellular contents into the cytoplasm, and ultimately it directly invaginates the lysosomal membrane, and there is budding of vesicles into the lysosomal lumen. While in case of the chaperone-mediated autophagy, the targeted proteins, the senescent proteins or the unfolded proteins, these are translocated via the LAMP2A gene into the lysosomal vacuole. So in these two processes, there is no formation of phagophore. So what are the applications of, where do we find autophagy? Now, in case of cancer, the autophagy can promote both cancer growth and also act as a defense against cancers by eating up the cancer cells. In neurodegenerative disorders also, there can be dysregulation of autophagy. Like in Alzheimer's disease, there is a misfolded protein. So if there is impairment of the autophagosome formation, this will lead to accumulation of the misfolded proteins, which can lead to the cell death and cell injury. Huntington disease, that is also a case of misfolded protein disorder in which there is mutant huntingtin. Then infectious disorders. Now pathogens, some pathogens are degraded by autophagy, especially mycobacteria and Shigella and herpes simplex virus 1. This macrophage specific deletion of autophagy related gene. Now certain genes are also important for autophagy. The deletion of this autophagy gene 5, this increases susceptibility to tuberculosis. And also there is application in case of inflammatory bowel disease. It has been seen that there's nucleotide polymorphisms in autophagy related genes like 16L1. This leads to the Crohn's disease. Now coming to some other vacuole forming modes of cell death. These are not that important for your exam, just for the sake of completion of your topic, and you can know this. One is NETosis. NETosis or cannibalism, that is characterized by cell-in-cell formation. Now, one of the cell, this interacts with other cell and cannibalizes the cell. In this, the molecule which is used is the Rho and ROCK. In this, either the cell will survive after the cannibalization or there will be internal cell death. Now, other modes, another is the MORPHOLOGIC. This MORPHOLOGIC is characterized by macropinocytosis, In this, there is formation of a large vacuole inside the cell, which cannot even fuse with the lysosome. So ultimately, because of the formation of this large vacuole, there will be a lysis of the cell, and the molecule which is implicated is the RAC molecule. Pyroptosis. In this also there is extensive cytoplasmic vacuolation which is derived from the endoplasmic reticulum or mitochondria, and the molecule which is implicated is insulin-like growth factor 1 receptor and the MAP kinase pathway. There's also some mitochondrial-dependent cell death, that is mitoptosis and parathanatos. The key molecules in case of mitoptosis is BAX, and they is characterized by mitochondrial disappearance and decomposition of the mitochondrial reticulum to small spherical organelles. In case of parathanatos, the key molecules are PARP and AIF, and in this also there is membrane rupture, the mitochondrial membrane permeabilization, chromatin condensation, and large-scale DNA fragmentation. Now coming to autolysis, autolysis is self-digestion. The term autolysis is usually applied post-mortem, means when the organs are out from the body, and in which there is complete absence of the surrounding inflammatory response. What happens is that the cell is disintegrated by the release of its own enzymes. So when the organs are out from the body, the there's cell injury, and the cell releases several enzymes which digest the own cellular contents. So in this we see this ghost outline of cells, and there is also superadded bacterial infection. Now, in some of the organs, this can be rapid, which are rich in hydrolytic enzymes like in pancreas and gastric mucosa. It is intermediate in tissues like heart, liver, and kidney, and slow in case of fibrous tissue. Now, the point of discussing autolysis is this. Now, after OT, the organs are sent to the pathology lab to see what is the diagnosis. So what we tell them is to fix the tissue in glutaraldehyde or formaldehyde. Now this fixation arrests this enzyme, this autolytic enzymes. This preserves the cell morphology. While if the cell is not kept in a fixative, the organ this will release various lytic enzymes which will cause damage to the tissue, and nothing will be visible at all. So for cell preservation, it is important to keep the organ in a fixative. So this is all about autolysis. So this is all about cell death. You should know the modes of cell death, and importantly you should know necrosis and apoptosis, and also the newer modes like necroptosis, pyroptosis, NETosis, and autophagy.